So welcome back, students. We will just revise the life cycle of Uchararia bank of the. As said earlier, this Uchararia bank of the completes its life cycle in two hosts, that is a mosquito and man, a vertebrate host and an invertebrate host. The invertebrate host, it is a mosquito, which is a vector, and a vertebrate host, that is a human. This host, since they completed life cycle in two different hosts, they are called as a digenetic parasites, and the parasite resides in the human lymphatic system. So the first, the larva, it is a microfilaria, and the microfilaria which are present in the circulation, what happens to that they actually migrate to the deeper circulation area, deep and the peripheral circulation. They usually migrate between the deeper circulation and the peripheral circulation of the blood vessel. Then during the daytime they are present in the deep veins, and during the night time they migrate to the surface blood vessels. That means they reaches the peripheral blood vessels. Then when the mosquito bites, when a mosquito bites an infected man, this microfilaria enters into the body of the mosquito, and in the body of this mosquito, what happens is that the microfilaria matures into a motile larva called as a juveniles. And when the mosquito vector has its next blood meal, these that is when this infected mosquito bites another healthy person, the juveniles are passed by the mosquito proboscis into the bloodstream of a new human host, and then the larva moves into the lymphatic system. Then it gets accumulated in the lymph nodes, predominantly in the leg region and in the genital area, and the larvae develop into the adult worms over the course of a year, and they reach the sexual maturity. And what happens is that after mating they attain the sexual maturity through its journey, or while its journey from the lymph vessels of the peripheral region to the deeper region, they mature, and the male and the female attains a sexual maturity, and through its travel they undergo the mating. Then they produce thousands of microfilaria. Then they migrate into the bloodstream. From the lymphatic system they enters into the bloodstream, and when a mosquito vector bite the infected human host, they ingest the microfilaria, and thus they repeat the life cycle. So again this gets repeated. So once once the mosquito gets infected, it transfers the microfilaria or the juveniles into the new human host, the healthy human. Then in the due course of time they gets matured, and when the blood is taken in by the another mosquito or the same mosquito, the microfilaria reaches the mosquito, and again the cycle gets repeated. So in certain regions the lymphatic system gets blocked with the microfilaria, and this results in the swelling of the lymph nodes, and in turn it can leads to elephantiasis. So the cycle gets repeated like this. Then it results in this lymphedema, that is the blockage of the lymph nodes, then elephantiasis, hydrocele, and these are the few pictures giving the infection caused by this microfilaria. Then when we pass on to the epidemiology, it's actually an epidemic in India, China and other countries. It is widespread, and then the infected person with circulating microfilaria are the chief sources of reservoir and infection, and from man to man transmission takes place by the bite of the mosquito. Then what happens is that actually this mosquito have the mosquito vectors of Wuchereria branchiophtha have a preference of human blood, and it appears that the humans are the only animals naturally infected by Wuchereria branchiophtha, and there is no reservoir host. Actually there is no reservoir host, and the microfilaria produced by the female worms have the membrane sheet, and this sheet along with the area in which the worms reside makes identification of the type of the species of microfilaria in the human easier. And these microfilaria are found mainly on the peripheral blood and can found to the peak amounts from 10 p.m. to 2 a.m. and at this time if a mosquito bites an infected man, this microfilaria enters into the body of a mosquito. The cause of this periodicity remains unknown, but the advantage is that microfilaria being in the peripheral blood during these hours may ensure that the vector will have the higher chance of transmitting them elsewhere. So the reason why there is a migration of this microfilaria from the deep regions into the peripheral blood vessels is still unknown, and after the night time it reverses. And there are the certain physiological changes associated with the sleeping, such as a slow body temperature and adrenal activity and a high carbon dioxide tension. And if the host sleep by day and is awake by night, the periodicity of the worm is reversed. It is reversed. So actual reason for this reversal is still unknown, but it is true that the periodical reversal is taking place. Then about the diagnosis of this infection by Wuchereria branchiophtha, there are the direct evidences are there, then there are the indirect evidences are also there. Direct evidences and indirect evidences are the direct evidences is the presence of this microfilaria. That is, if you take a blood smear, we are able to diagnose the presence of this microfilaria. We are able to find these juveniles in this blood smear, and sometimes the infected people do not have the microfilaria in their blood, and as a result the test aim to detect the antigen test are used. If we could not find any microfilaria in the blood sample or the blood smear test, then you can go for an allergic test, that is a blood examination is done. Then as a result the test is aimed to detect the antigen from the adult worms can be used. Then ultrasonography test can also be done to detect the movements and the noises caused by the adult worms. Then with the help of the X-ray, the calcified worm can also be identified. Then other tests are the biopsy, then serological diagnosis such as ELISA and RIA can be done. Drop biotest can be done. PCR assay for the detection of the microfilaria infection can be done. Then X-ray examination, which shows the calcified adult worms, is also possible. So these are the various measures by which the infection of Wuchereria branchiophtha is detected. Then about the treatment, the severe symptoms caused by the parasites can be avoided by cleansing the skin Then surgery or the use of the therapeutic drugs such as diethylcarmazine can be used. Then albendazole can be used, ivermectin. These are the other drugs which can be used for the treatment of this Wuchereria bancroftyi. Most widely used drug, it is diethylcarmazine. It is widely used for the treatment against this microfilariae of Wuchereria bancroftyi. That is that we can eliminate the microfilariae from the blood and also kill the adult worm with a dosage of 6 mg per kg, with a 6 mg per kg semi-annually or annually. We can use this drug according to this dose, annually or semi-annually, and this infection can be checked. Then this cheap and effective, this medicine, it is cheap and effective and safe with the new side effects such as a fever, chill, headache, etc. So dosage can also be given in this means, that is the first day we can give the 50 mg after food, then second day 50 mg 3 times daily, or the third day 100 mg 3 times daily, and the fourth day. Then on the fourth day to 21st day 5 mg per kg per day in 3 divided doses. So these are the 3 divided doses through which we can give. So this varies according to the infestation rate, or the rate of infestation. The dose also varies. Anyway, this drug diethylcarmazine, that is DEC, it is widely used for the treatment of this Wuchereria bancroftyi. Then about the prevention and control, the prevention focuses on the protecting against the mosquito bites in the endemic region. So this mosquito can be controlled so that the vectors can be prevented from transmitting this disease to the new host. Then the clinical control of spraying the insecticides, DDT, malathion, etc. to control the mosquito, or biological control can be used by using the bacillus species. Carnivorous bacteria can be used to kill this vectors. Then carnivorous fishes can be used. Then environmental control by the efficient drainage and the sewage system can be used to prevent the mosquito from flourishing. Then there is a chemotherapeutic control can be done, that is by using the certain clinical methods. The reduce morbidity due to the filariasis by treatment the clinical cases. Then lower transmission by treating the case of crofilariemia, that is the filarial infection can be controlled by certain chemical therapies. Then interrupt the transmission of the infection. Then based on the mass or the selective treatment of the cases by administrating DEC, that is diethylcarmazine, can be administered at periodic interval, and this infection can be checked to a great extent. So basically the vectors can be controlled, and thereby we can check the infection caused by Wuchereria bancroftyi. And if once infected, or after infection or before infection also, we can just apply some therapeutic methods, that is mainly the DEC is given at the smaller dose for every individual, and we are able to check the spread of this nematode parasite. So you might have heard about the deworming procedures undergone by the government in order to check the spread of this infection caused by the nematode parasite. So deworming is a common procedure done along with the vaccination. That's all in this video.